Morning, sir.
Good morning. See. Okay. I'll try to finish by 10:50. I have a meeting that's I'm out of a meeting actually, so I want to go back at 10:50, 10:50. So I'll try and finish it if possible by 10:50. Okay, how are all of you doing? All of you doing good? Everybody keeping good health?
Yes, sir.
Yes,
Okay.
yes.
How are you, sir?
I'm good. I'm good. I got infected and I recovered. We went to
Many people at KSBS have been infected.
Yeah, mine was actually another case. We chose to go on a holiday in the last week of December to Port Blair and a couple of islands in Andaman, Nicobar, and probably got infected while having food in crowded restaurants. That's my guess. I don't know how I got infected, but we all did. Almost 12 of us who went on a trip got infected, all 12 of us. Okay, but thankfully we've all recovered well. Okay, today what we're going to do is we're going to look at virus classification. Let me grab a pen from this. Okay, now can anybody think of some virus names and think what are they named based on? Just based on whatever we've looked at so far in this course or anything else that you know, how are viruses named? Just think of a virus and think of its name. And what is the
Based on
basis for naming?
diseases and based on origins.
Based on origin as in origin can mean a variety of things. Anybody else?
So
Based on the site
based
at
on
which
disease, they affect. Like
Okay,
based on the disease,
based
they
on the
infect.
disease they could cause. That's correct. Somebody else was saying something.
based on the site at which they replicate,
Perfect. Based on the site that they can replicate.
and
Anything else
also
you can
based
think
on
of?
the system
Sir,
by which
based on their
Go ahead.
structure, sir.
All,
How they
all
look
are
like.
correct. All are correct, but how? I'll just give you some examples, some crude forms of naming viruses, but they stay. Okay, so the viruses could be named based on the disease they cause, like some of you rightly said. Poliovirus causes poliomyelitis, rabies virus causes rabies, HIV causes immunodeficiency. Like this virus, all of you heard about, we spoke about these viruses, Coxsackie viruses, and I tried to tell you these are suburbs on the outskirts of New York. So they could be named after the places or geographic region. They could be named after the scientists who discovered Epstein-Barr virus, for example. And a lot of viruses also have like Greek and Latin names, like influenza means influenza of bad air, which means that basically it's spread by air. They figured it out long time ago, but they just named it like that. Then dengue viruses means evil spirit. I don't know in which language, but they were named like this. But then if you were to think about it a little bit more carefully, and some of you already mentioned a variety of these, they could be named after the host that they infect, the target tissue that they infect, the route of transmission like HIV, HBV, and HCV, for example, are classically dubbed as blood-borne viruses. And all of you know enough examples of respiratory viruses these days. And there are arthropod-borne viruses, and they could be classified based on the genetic material or the structure. I think most of these things popped up in the discussion. Okay, a few more interesting things about virus classification. So the first thing that you want to recognize is that, I don't know how many of you know this, how many of you do not know this. I think everybody knows this. So this is, you have this classification that starts with kingdom, phylum, class, order, and then it goes to family, genus, and species. So in viruses, we don't have this part at all. We only have, it only starts at family, then you have genus and species combined. I'll explain that a little bit to you. Because see, there's plant kingdom and animal kingdom. We don't know whether viruses belong to any one of them. Then there are animals with backbones, animals without backbones, and things like that. That contributes to this part. So, but for viruses, it doesn't exist, simply doesn't exist. So there are about 20 virus families in all, and there is no classification of this level. For viruses, the highest level of classification for viruses is that it starts with a family name. The family name ends with Viridae, DAE. And then there are subfamilies and their names end with Virinae. Okay, I'll give you, I think I have an example, but for example, Picorna viridae is the name of the family, and then there are subfamilies within it. Okay, so Picorna viridae, we all looked at it, right? You all know about it. And then subfamilies, some viruses may have subfamilies and they end with virinae. Not all viruses have this, but all viruses, the name ends with something dash viruses like polio virus, rabies virus, okay, SARS-CoV-2. So everything ends with a virus. And this is actually the genus and species name combined. So is anything different here from any other classification that you know of? Guys? What is our biological name?
So there are different genus and different species.
That's right. We always have a genus name and a species name. This is called, what is that called?
Binomial nomenclature.
Binomial nomenclature, and there is no binomial nomenclature for viruses. Is not adapted as yet because people were not sure, and then it's never been adapted until this date. Like for example, humans are Homo sapiens, dogs can be Canis lupus, cats are Felis catus, and there are many species, but there are two names, right? All of you know that. Genus name followed by the species name, Escherichia coli, Klebsiella pneumoniae. So genus and species name, this nomenclature is not followed for viruses, and probably they are the only organisms in the whole world for which this is not yet adapted. Okay. Here's an example of what we commonly call as chickenpox virus. Its actual name is Varicella zoster virus. That doesn't mean this is the genus and this is the species. It does not mean that. Okay, it's just the genus name. Some people think it's genus / species name, but it's not genus name and species name separately. This is not genus, this is not species. It's not like that. It has genus / species name, only one name. It's Varicella zoster virus. And herpes viruses, so it's herpes viridae. This is the family. There is a subfamily in herpes viruses, so it's virinae. This is just an example so that you get it. There is alpha, beta, gamma subfamilies. This one belongs to alpha, and commonly we refer to this as chickenpox virus. Okay. I'm going to move on to historically the most important method of classification that emerged for viruses. It was done by David Baltimore, who went on to get a Nobel Prize, not for this work of classification, but for some other work, something to do with reverse transcriptases. Okay. So what did Baltimore do to classify viruses? He thought about how viruses replicates, and suddenly he recognized something that is very simple, but that became the basis of his classification. And historically, this was very important because it made the whole world understand how viruses replicate better. So what was so unique about Baltimore classification? Actually, it's a very simple method of classification. Today, you can think that this everybody knows this, but you have to think about it, then you have to classify it, then there is a lot of biology that comes with it. What is the basis of Baltimore classifying viruses? The one single point theme based on which he classified all viruses is that he figured out that viruses encode their own protein. So they should have their own mRNA. So whatever kind of virus it is in terms of the genetic material, they all converge at one point that all viruses make their own mRNA. And this is, I think if you ask me, or if you ask another person, or if you think about it, this looks like, yeah, of course I know it, but you have to think about it more carefully. Then a lot of things fall into place. That's what he did. So this is the central or focal point on which Baltimore classification was based on. So just just like I told you, his entire theme is based on all viruses make their mRNA. So let's start with this class here. This is double-stranded DNA viruses. You have one strand here, you have the second strand here. You can see that. Now, one of these two strands will serve as template and mRNA will be made. Now, there are single-stranded DNA viruses. Okay, so for single-stranded DNA viruses, how do they replicate? If they have to replicate, they have to make mRNA. If they have to make mRNA, they have to go to a double-stranded DNA step. This is an intermediate step. This is inside the virus. This is the only genome that's packaged. But when they are replicating inside a host, they have this as an intermediate step, which is the double-stranded DNA step. And from these two strands, one of the strands serves as template for mRNA synthesis. And so for any single-stranded DNA virus, they go through a double-stranded DNA intermediate step, which is a transient step, which happens inside the host. After this, one of these two strands serves as template for mRNA. Now, let's go on to the next class that he defined, which is double-stranded RNA viruses. So you have one strand of RNA in one color, the other strand in another color, two shades of green. Now, one of these two strands, in this case, this particular strand is same as mRNA. What does that mean? That means, anybody wants to answer that? Go ahead.
In case of double-stranded mRNA viruses,
There is no
naturally
double-stranded mRNA virus.
So what is this?
This is double-stranded RNA virus. It's not double-stranded mRNA virus.
Yes, sir.
M stands for message.
Yes, sir. Yes, sir.
Yeah.
Double-stranded RNA viruses. How long double-stranded RNA as a genetic material are found in viruses naturally?
I didn't understand the last part. How double-stranded
How
RNA?
long? Like maximum how long
How
can
long?
it be?
How long?
RNA
Okay.
virus. Yeah.
Okay. Double-stranded RNA viruses are somewhere around 10 to 20 kb. Most of the ones that are known are in that range. Is that clear?
Yes, sir. So are they? How are they like? They form an stable genetic material.
How they form
RNA is
stable?
stable. I think.
Yeah. So most viruses that we know of are RNA viruses, including our pet SARS-CoV-2, and it is genetically stable, and that's why you and me get infected, right?
Okay, sir. Thank you, sir.
What is your question? Is RNA genetically stable?
No, sir. Like double-stranded RNA.
Double-stranded RNA is nothing too different. See, I'll come back to it in 1 minute before I end this slide, so that you can appreciate there's nothing very unique about double-stranded RNA. There's only one enzyme that will be needed for all RNA viruses, that is RNA-dependent RNA polymerase. And sometimes, like some viruses are single-stranded DNA, some are double-stranded DNA. Just like that, some viruses are single-stranded RNA, some others have two strands together as the genetic material, and they are very stable. I mean, by stable inside the capsid, they are good. Actually, they don't disintegrate very easily. Yeah, go ahead. Somebody else?
Yeah,
Yeah.
actually, I think he wants to ask that RNA is thermodynamically not stable because of that 2' wedge present in that ribose sugar. So in that case, how they like survive after replicating like after infection? I think so. So
So
in that case, they convert into complementary DNA, cDNA. Then they pursue their.
no,
Is it like
actually,
that?
that is not correct. Actually, most RNA viruses do not have a DNA replicative intermediate step. Most of them don't. Some do, most don't. For example, SARS-CoV-2 does not have, influenza does not have, poliovirus does not have a DNA intermediate. They don't have a DNA equivalent at all in the system, anywhere, inside the host, outside the host. They don't have anything to do with DNA at all. See, why is RNA unstable? Generally, because there are plenty of RNases that can be there on the table, and it can be inside your body, inside your cells. So RNases are there in plenty. So that's why the RNA that we make from our cells, it has a 5' guanosine cap and a 3' poly A tail, which makes it more resistant to digestion by these RNases. These two are important for our RNA. Like these viruses, many viruses can also have 5' caps. They can also snatch the cap from our host mRNA. They can do a variety of things. And many virus RNAs also have a poly A tail. When they make transcripts, they also have a poly A tail. Many virus RNAs also have that. But regardless of that, see, the only reason RNA is less stable is primarily because of the existence of RNases. And the only other difference between DNA and RNA is that RNA is more susceptible to alkali pH. So alkaline pH RNA degrades faster than DNA. Other than that, pretty much they're not too fussy, or they're not too fragile like generally thought about. They're quite stable. Okay, is that does that answer?
Yes, sir.
Okay, I'll tell you one more thing so that you can relate to this double-stranded nature of this RNA shown here. But there are some viruses which have two strands of RNA as the genetic material. But we'll come back to this. 1 second, I'll give you an example so that you can relate to it better. Now, you have negative sense RNA viruses, and you have something called positive sense RNA viruses. Now, can SARS-CoV-2, what category do you think it belongs to?
Single strand RNA.
Single strand RNA, but do you think it belongs to this category or this category? I'll tell you the difference in a second.
So positive,
Yeah,
positive
it belongs
single-stranded
to this
RNA.
category right here. You're right. And influenza viruses, for example, belong to this category right here, negative sense. What is the difference between positive sense RNA and the negative sense RNA? So what it means to when they say like SARS-CoV-2 is a positive sense RNA virus, what that means is that the 30 kilobases of genetic material that it comes with, that is packaged into the virion, that can be directly translated into mRNA, or that is same as the messenger RNA for the virus. The genome is equivalent to the messenger RNA, or it's the same as messenger RNA, which can be converted to protein. Or in other words, the SARS-CoV-2 genome, when it enters a cell, it can serve as template directly to make SARS-CoV-2 proteins. So that kind of genomes, where the genome itself can act as a template for protein synthesis, or the genome itself is equivalent of mRNA. Like you can see, this color and this color are same here. So when the genome itself is equivalent of mRNA, or it can directly translate into proteins, you call those as positive sense RNA. Okay. Now, what is negative sense RNA? The genome of these viruses, which are negative sense RNA, they are complementary to the mRNA. They're complementary to the message that can be translated to protein. What does that mean? That means that they will. So this is, for example, let's say influenza virus. This is the genome of influenza virus. Now there's going to be an enzyme that is going to be RNA dependent RNA polymerase that copies this, and then you get a complementary strand for this. Now this complementary strand right here is same as that of the mRNA, which means that the genome is not equivalent to mRNA, and the genome cannot be directly translated into virus proteins. Then that class of RNA viruses are referred to as negative sense RNA viruses. Is that clear to everybody?
Yes,
Yes?
sir.
Yes, sir.
Okay. Now, if you think about,
I have one
yeah,
question.
yeah, go ahead.
So now we have two types of RNA. Sorry. Some are positive strand and some are negative strand.
You
So
call it strand, you call it sense, SENSE, sense.
so one thing you have told us that these positive RNAs are equivalent to mRNAs.
True.
So the positive sense RNA and the negative sense RNA, do they have some advantages and disadvantages to make them more effective or some other advantages?
Yeah, okay, so that's a good question. Um, let me put it this way. I'll finish the next step and then we can talk about that same thing. There are two things we'll talk about after I finish one more small thing. See, if you look at it, let's say this is the mRNA. Okay, it can be directly translated into proteins like I've shown you already. But typically what happens when this gets into a cell is that it synthesizes a negative sense RNA. So it synthesizes complementary RNA. Okay, so this complementary RNA is now going to serve as template for the virus to synthesize plenty of mRNA or plenty of genomic RNA, both of. So this is an intermediate that will allow the synthesis of genomic RNA, which is same as mRNA in this case. I'm just going to, it's becoming too crowded, even I can't follow it. Okay, so if this is SARS-CoV-2, this will be the intermediate. I'll just put I for intermediate. And this serves as template for the synthesis of more genomic RNA, which is packaged into SARS-CoV-2. And this is same as mRNA, the same thing. This will also serve as the template for the synthesis of all the necessary virus proteins. So this is an intermediate here. Now look at this case here, negative sense RNA. So now for negative sense RNA, what it does is it will synthesize a positive sense RNA, which is the mRNA. This positive sense RNA has two functions. One, it serves as template for the synthesis of the negative sense RNA. See, they're complementary to each other. So this will be template for the synthesis of negative sense RNA, which is also equivalent to the genomic RNA, which gets packaged into the virion. Now, this also serves another function. This positive sense RNA, which is the intermediate, will be translated into virus proteins. So if you look at it, there are some advantages possibly, and there are some disadvantages. But for whatever reason, I think that people have studied it. They think actually these viruses, negative sense RNA viruses, some of them are associated with high mortality, but we don't understand the exact reason of their association with high mortality rate. But that could be possibly related to something to do with the immune response they generate or the way they replicate. But the point I wanted to make is that be it positive sense RNA, like the one that I'm going to circle now, or be it the negative sense RNA like this one here, it always makes the other strand inside the host, which serves as template for different things. It may serve as template for mRNA, it may serve as template, it may actually be the mRNA itself. So here it serves as template for making mRNA, and this one here is the intermediate, but it is the mRNA itself. So pretty much no matter what the class of RNA is, they always have an intermediate with the other strand. That is a must for all RNA viruses, either positive sense or negative sense. Now, there could be some advantages for positive sense RNA viruses, just because one of you asked that question. That's a good question. So some advantages for a positive sense RNA virus. I'm just going to clear like this one right here. What advantage could it potentially have? Let's say this is SARS-CoV-2, it just enters a host cell. And viruses, many viruses, not all viruses, but many viruses have something called temporal production of proteins. What does that mean? I'm just writing T for temporal. So many viruses, they don't produce all the proteins at one go. They don't. So they produce some proteins called early proteins, some proteins called, uh, what are they called? 1 minute. Immediate, yeah. I'll just reclassify that. They're called immediate early. The second one is called early, and the last one is called late. This may be produced within, let's say, 5 hours of the virus getting into the cell. This may be produced, let's say, 20 hours after the virus gets into the cell. And this may be produced like 35 hours after the virus gets into the cell. And these are usually the last structural proteins that are necessary for packaging and release. This class is that. This class here may be something that is necessary for the virus to replicate. For example, RNA-dependent RNA polymerase that the virus brings along with it, that the virus needs immediately after getting in. So for many of these viruses, this kind of temporal production of proteins exists. Not all, but many. We don't even understand that well for some viruses. It's only studied well enough for some viruses. But regardless of it, many viruses don't produce all the proteins at one go. And this way, if something needs a protein as soon as it enters the cell, perhaps this virus right here, this class of viruses which have positive sense, because they can immediately translate whatever genome enters there, that can be translated into virus proteins, at least the parts that they want, it could be done immediately. So these may have a marginal advantage in that sense. Does that make sense to you?
Yes, sir.
Okay, that is,
Yes, sir.
there is actually a lot of intricacies here, which I cannot discuss in this class because of time and I have to finish this. But there is a lot more unknown also, but the question is actually a good one because there could be something associated with it. That's why I gave you the example that this class of viruses are generally among the most fatal human viruses. Many of them belong to this class. So we don't know why. It could be something to do with immune response. It could be something to do with how it replicates. It's worth investigating, but we don't know enough yet. But this is a direct thing that I told you. Is everybody clear with that?
Yes, sir.
Yes, sir.
Okay.
Sir, actually, I have a doubt.
Yeah.
In positive RNA or the sense strand containing virus. So actually, when they infect, they hijack the host transcription system, that is the RNA polymerase or anything. In that case, like how this positive RNA immediately translate into protein, like in that case, there is no requirement of RNA polymerase of their own, like which they contain in this RNA. So
So
what are the
okay. So,
immediate proteins? Yeah.
no. What you want to understand is that even this class of viruses, although I just told you that this can be translated into proteins immediately, what you said is you think that they will not require an RNA polymerase because they can already make the protein. Is that your thought?
Yes, because it is
That's
something
not true.
related.
I'll tell you why that doesn't work. See, when a virus gets into a cell, it replicates, not it does not double like bacteria. Pretty much most organisms that we know of, like including human cells, one cell becomes two cells. That's how your hair, skin, everything grows. right? 1 becomes 2. Same thing is true for bacteria. So this is called doubling. Viruses don't have doubling. What they have is something called a virus replication cycle where there is exponential growth. What does that mean? When one SARS-CoV-2 virus, for example, I'm just using that example because all of you are full of it and can relate to it easily. Nothing more important about this virus otherwise in terms of virology except the public health point. Uh, if you look at it, let's say one virus infects a respiratory cell. How many viruses come out? Okay, like 10,000 to 100,000 viruses can come out of the cell. That is the way most viruses normally replicate. One gets in, 10 to the 5th, 10 to the 6th number of viruses come out. So if this happens, if one gets in and if you don't have RNA polymerase, you'll be left with just one genome that can be packaged. And packaging and secretion is an inefficient part of virus replication. You need, if you have 100,000 RNA molecules and 100,000 capsids and 100,000 envelopes made, maybe only 1,000 or 5,000 or 10,000 will be packaged and they will come out. It's an inefficient process. Not everything can find the, it's not a 1:1 thing and it's not a perfect system. So here, if you don't have RNA polymerase, if you cannot go into this step, you'll be left with one genome, and one genome, maximum you can have 0 to 1 viruses. Most likely it will be 0, the virus cannot replicate. So that's why it needs the intermediate. So all virus, so this needs this, and this class of viruses need this. So without these intermediates, nothing will happen. Does that answer your question about why the intermediate is needed, or why RNA pol may not be needed? It is always needed because of this reason.
Yes.
Give me 1 second. Hi, Himani. Sorry about that. Is this clear to everybody?
Yes, sir.
Okay, so I'm just going to go on to
Sir, I have a question actually.
go ahead.
Sir, extending to last question, like he said that these viruses hijack the host machinery of RNA polymerase,
All
right?
viruses hijack host machinery. There is no
No,
go ahead.
so RNA polymerase in host are DNA dependent, but
Yeah.
they
Actually,
require RNA dependent RNA polymerase, right?
yeah, that is not correct. So all RNA viruses require their own polymerases. That is a simple statement.
Okay, so they carry their own polymerase.
That's right. Sometimes it can be packaged along with the virus. Sometimes it may not be necessarily packaged along with the virus, but that is the difference. The protein may be packaged with the virus or may not be packaged with the virus, but the genome always encodes RNA polymerase, and it's a must for all RNA viruses because we don't have good RNA-dependent RNA polymerases. We do have, but it's made under special circumstances.
Okay.
Does that answer your question? So all RNA viruses bring their own polymerases. Actually, that's true for pretty much most DNA viruses also.
Yes, sir.
Although we have DNA polymerase, they bring their own polymerases. Okay, now let's go on. So there could be RNA viruses that replicate with the DNA intermediate. So one example of this class of viruses is a famous virus that all of you know. It's HIV. So what is this step? This is going from RNA to DNA. So what kind of step is this in the central dogma?
The reverse transcription.
Reverse transcription.
So these viruses are called retroviruses or reverse transcribing viruses. So HIV is a virus like that. It goes into DNA and then it becomes double-stranded DNA, and then double-stranded DNA serves as template. It goes back to yield this. So this is same as this, right? So it goes back to yield the genomic RNA also and mRNA also, and then the virus replicates like this. So the bottom line is that whatever be the virus type that you have, they all converge at one step called the mRNA, which is virus's own mRNA or messenger RNA necessary to produce the virus protein. So this was the basis of Baltimore classification. We don't use it because many unrelated viruses will fall together. Now, subsequent to this, there was another type of classification that where they classified the virus nucleic acids, the type of capsid, whether it has an envelope or not. And again, the problem with this kind of classification. Yeah, go ahead.
Sir, you were saying that many unrelated viruses will fall together, sir. How come that the viruses may have an evolution? So how come these viruses evolved? If they evolved if there is a single-stranded positive mRNA virus, then that may have been evolved from some similar mRNA virus and not from something like double-stranded DNA virus. So how come they be
Yeah.
the
Yeah.
Baltimore's classification be irrelevant in that sense?
No, it's not irrelevant like that, but it's all in the understanding of what we define as what are unrelated viruses. Give me a second. I'm so sorry. Sorry, guys. Something to do with some medical situation. Don't sorry about it. Okay, so where are we? What is the question? What is the question?
The question was related to evolution of viruses and about like how these viruses are unrelated, something like that.
Yeah. Okay. Okay. I got it. Okay. So the answer is actually it all depends on the perspective of what we call unrelated. So something like for example, something that infects your causes diarrhea, something that causes respiratory tract disease, and something that causes a chronic disease, and they are completely unrelated sequences. They all fall into one class of viruses. I'll give you an example. Maybe I can show that to you here. See, if you look at it, this is flaviviruses, is influenza virus, toga virus, toga viridae. There are variety of viruses. Coronaviruses are this class of viruses. So influenza virus. So there are a variety of things that 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 can be in flaviviridae is not influenza. Orthomyxo is influenza, but there are a variety of diseases and unrelated sequences that can come in the same class. I'll give you one example. So maybe this is influenza virus actually, this family. This is closely related to influenza virus. This is rabies virus. This is Ebola virus. They don't have anything in common. If you look at it, these two do, but the others don't. Rabies and influenza, respiratory tract, brain. Filovirus, Ebola and Marburg, they have 50 to 90% mortality. They have completely different. So they don't have anything to do with each other in terms of sequence, the way they cause disease, the disease they cause, the organs they infect, but they come under the same family. That is what it means. Is that clear?
Yes, sir. Is it the case like their evolution has happened with the same family in terms of like David Baltimore's classification?
No, he does not say anything about evolution. He just says that if you put all these viruses together, you can call them one class because they have this as intermediate. If you put all these viruses, they have this as intermediate. So he's not talking anything about evolution. He's just saying that they belong to a certain kind of replication mode.
Yes, sir. Yes, sir.
Is that clear?
Yes, sir. Clear.
Okay. So this also is not good because even if you look at the type of nucleic acids and whether it's enveloped or not, and look at the type of capsid, all these things done also, then also unrelated viruses may end up in the same family. So what do we use? We use today something that is based on the DNA or RNA sequence, and based on how closely they are related, there are over 5,004 to 5,000 viruses. Now I think this is 73 families are described as like until a couple of years ago. So they are classified into a system like this based on a virus family, subfamily, and then there is further classification beyond that. I'll tell you what that is in a bit. But they are based today, the virus classification system that we have is completely based on the virus sequence and how closely they are related. That's what we are using today. This is the one that's currently in use. Now, I just went through the slide. You don't have to by heart anything here. These are just some types of classification of viruses that is followed, but you don't have to memorize any of this. This is again something like that, where it just gives you an overview of what are the groups of viruses. This is like if you look at the Baltimore class, this will be one class. These are the different hosts, how they infect, like Rhabdoviridae can infect a variety of hosts, things like that. This is just a cartoon I picked up from somewhere so that you get a sense of how widely they're distributed. Now, I think we'll have to stop here. I was going to finish another 2 or 3 slides, and this may be more interesting than just the naming, but I think we'll have to do this in the next class. We'll do this on Friday. If you have any questions, guys, tell me. Any questions?
None currently, sir.
Sir, would you like to suggest some book for studying this viruses or some other kinds of study material?
Okay. If any of you are interested, you can email me. I have a book by I'll also flash it in the next class if you remind me. It's the authors are Javits and Melnick. It's called a textbook of medical microbiology, and this is standardly used for teaching medical students and anybody who wants to do human virology. This is a simple but a good book. It's called, the authors are Javits and Melnick. And there is also one more author who I can't remember. We just call it Javits. I have the book if you want to borrow it. But the gist of it is all these things you'll find, but like I think
Yes, sir.
most.
Then I can take. I am here.
Yeah, you
So
can call me or you can email me and coordinate, and you can pick it up if you want.
yes, sir. I will email.
Okay. Okay. Okay, then we'll meet in the next class then, Friday. Bye.
Thank you, sir.
Thank you.
Thank you,
Thank
sir.
you, sir.
Thank you.
Thank you, sir.